So if this were a agree-disagree question on the next exam. Oh, very good. Would you agree or disagree with this? How many would agree? Anyone disagree? How come you would disagree, Rick?
As in terms of life on Earth, it would be the most important. But
Okay.
I'm thinking like the formation of the planet might be the most.
Okay. That would be a different tack you could take. Here's kind of my logic. There are several things that are relevant to photosynthesis and importance. And one of the big ones is that it really, for the first time, allowed the Earth to be an open system. Remember what an open system is? An open system is one that can exchange energy and matter with its surroundings. Okay? Now, we were getting energy from the sunlight prior to the evolution of this process of photosynthesis, but we couldn't use it. Organisms could not use it. So in that regard, energy-wise, Earth was really a closed system. The energy that evolving organisms were using were materials that were on the planet. Okay? And if they got used up, life would have stopped. And so this ability to tap an outside source of energy, to have producing organisms, was really crucial for the sustainability of life on Earth. Other big thing, oxygen was produced. Okay? We talked about the significance of oxygen in a couple of contexts. Number one, if you have oxygen, you can have ozone. And the development of the ozone layer allowed this transition from aquatic environments to terrestrial environments. And that was huge in terms of kind of the development of life on Earth. And with oxygen, you could now have aerobic cellular respiration, which hopefully you'll appreciate after the last exam is a lot more efficient than fermentation. And aerobic cellular respiration was tied up very closely to the evolution of eukaryote organisms. There are very few eukaryotic organisms that don't have the capability for aerobic cellular respiration. Also, two of the major organelles of eukaryotes we think arose by endosymbiosis, and those were the mitochondria and the chloroplasts. Okay? We're not going to have mitochondria unless we have oxygen. And then finally, the evolution of multicellularity and having organisms that were more than one cell is also tied up with this ability to produce significant amounts of energy. And we are, I guess, the culmination of multicellular evolution. So there's a lot of things that photosynthesis caused, directly or indirectly, that were pretty important in terms of the evolution of life on Earth. So I think it's fitting that we spend some time talking about that. Remember that last time we were breaking up the reactions of photosynthesis into kind of two sets of black boxes, the light-dependent and the light-independent reactions. Where do the light-dependent reactions take place? They take place in the thylakoids, which are the membranes of the chloroplast. Where do the light-independent take place? They take place in that region called the stroma. So the chloroplast is organized in such a way that it has different locations for these particular processes. What do we need for the light-dependent reactions to take place? Light, okay. And if we have sunlight, we also need something to do what to that sunlight? Absorb it. Okay, so we need light. We need pigments to absorb it. We need some membranes, okay? Membrane-associated process. What are the other important reactants of photosynthesis? Okay, carbon dioxide. What's the other one? Water. Okay. Which of those are used in the light-dependent reactions? CO2 used? Yeah, water is used. But carbon dioxide isn't going to be used in the light-dependent reactions. That's going to be something that's involved in the light-independent reactions. So what's the critical event? Well, the critical event is that when light strikes these pigments, electrons start moving around. So we get electron transport. And with electron transport, we can get an H plus gradient. And what's the H plus gradient allow us to do? Make ATP, right? We have chemiosmosis. What happens to the water molecules in the presence of sunlight when pigments absorb that light energy? They're broken apart, aren't they? And we actually break those bonds, those covalent bonds. And what do we generate? We generate atoms of oxygen and hydrogen. What do a couple of oxygen atoms do? They bond with one another to form that waste product, molecular oxygen, which was so important in the evolution of life on Earth. What happens to the hydrogens? Well, they're picked up by a coenzyme, and that coenzyme becomes reduced. So, our major products of this are these three things right here. These forms of chemical energy are then used in the process of, we call it carbon fixation, but CO2 is actually being reduced into the chemical energy, glucose, that can be then used in cellular respiration. Remember what another name for these light-independent reactions are? This particular process is called the Calvin cycle. So, there's a good kind of visualization of the relationship between the light-dependent reactions and the light-independent reactions. And one of the things that I showed you the other day is that these particular thylakoid membranes are organized in such a way that the thylakoid membrane has a space inside of it. And so we have a setup where a membrane is separating one compartment, the space inside, the thylakoid space, from a second compartment, the stroma. And remember that a membrane separating two compartments is a prerequisite for setting up a hydrogen ion gradient, and you need that hydrogen ion gradient to do chemiosmosis. Okay, here's a question for you. Once we form glucose, then you can run glucose through cellular respiration and make ATP. Why do we need this process occurring in plant cells if the light-dependent reaction's producing ATP? Why can't plant cells get by with just photosynthesis? Why do they need cellular respiration as well? There's a real obvious answer and then a less obvious answer. What's the real obvious answer? Light's not always shining, is it? Okay, what would happen to plants at night then? If they stop producing ATP by this light-dependent process, remember we call that photophosphorylation as opposed to oxidative phosphorylation? That's fine when the light's shining, but what do they do in the dark? Another less obvious answer is it's really important to produce glucose because is the only purpose of cell respiration to make ATP? Last exam question. What's another important purpose of cellular respiration besides making ATP? That anabolic process, remember, making those carbon skeletons? Plants, once they make glucose, if they don't need to break it all down to make ATP, can make lipids, proteins, nucleic acids, all the things you need to make and repair new plant cells to sustain growth. Now, sometimes you need some additional materials to make these molecules out of sugar because nucleic acids and proteins both have nitrogen, right? Does glucose have nitrogen? No. So where do plants get their nitrogen? From the soil. Yeah. So they have to obtain some additional nutrients in order to convert this glucose into these other molecules that make up cells of living organisms. The primary photosynthetic organ of the plant is obviously the leaf. And leaves have some adaptations that allow them to be very efficient in terms of this process of photosynthesis. One of the things that leaves typically are characterized by is being kind of broad and flat and pretty thin. What's that an adaptation for? Increasing surface area for absorbing more light, right? If you look at leaves in a cross section, what you'll see is that inside of a leaf there is this sandwich of cells basically in the middle. And these cells are very rich in chloroplasts because that's where the pigments are that are going to absorb the light. This tissue in the middle of the leaf is called the mesophyll. The prefix meso means middle. Okay, phyll refers to leaf. So this is this tissue in the middle of a leaf. Now, for photosynthesis to occur, we do need some water, right? And for those leaf cells to be turgid, and therefore for that tissue to be erect and not all shriveled up, that requires some water, right? So we're going to need to be able to bring water from the soil up into the leaves. And so there's a plumbing system. There are some veins in the leaves. There are actually two kinds of plumbing tubes in plant that carry materials necessary for plants to function. What are the tubes called that transport the water? Remember? They're called xylem, X-Y-L-E-M. And xylem, another word for xylem is wood. That is the woody tissue of the plant. Another thing that plants need to be able to transport, particularly from the leaves back down to the roots, are the sugars that are made during photosynthesis. So that food is transported in another kind of plumbing system. Do you know what that one is? That one's called phloem. So we have this xylem and phloem, which are organized into these transport tubules that we call veins. Okay? The upper and lower level of the leaf is called the epidermis. Epi means outer. Dermis refers to skin, basically. So that's the outer skin of the leaf. And in the lower level of the epidermis, in the lower layer of the epidermis, there are these little holes in the leaf that can open and close. There's some specialized type of cells called guard cells that surround these little holes. And when they inflate, the holes close up. When they deflate, the holes open up. And what these particular structures do, the stomata, is they allow gases to be able to move across that epidermis. Why might gases have a difficult time moving across this epidermis? Well, leaves have a problem. And one of the problems that leaves have is that because they've got a high surface area, they tend to lose water very readily by evaporation. And if all the water evaporates out of the leaf, what's it going to do? It's going to shrivel up. It's going to wilt. Okay? And then it's not going to absorb sun very well. And then it's not going to have the water it needs for photosynthesis. So it's really important to keep the leaves hydrated. So what plants do typically is they secrete this waxy substance that accumulates on the upper and lower epidermis. That waxy layer is called a cuticle. And it's great at keeping water in the leaf. But it would prevent then oxygen from leaving the leaf, and very importantly, carbon dioxide from getting in. Okay? Carbon dioxide's got to enter, over here, the leaf for these light-independent reactions to occur. You might say, well, is it really that important that oxygen get out of the leaf? Because isn't oxygen going to be used for cellular respiration? Yeah, some of it. But plants can produce way more oxygen by photosynthesis than they might need during respiration. And the oxygen can build up. Is that a problem? Yeah, actually it can be. That's something we'll talk about later on, is that too much oxygen can actually inhibit some types of photosynthesis. So it's very good to have a mechanism to get excess oxygen out of the leaf. So these stomata can open and close depending on kind of the needs of the plant. And if it needs to conserve water, like on a hot, windy day, those stomata might close down for the most part. Well, that keeps the leaf hydrated, but it's going to prevent CO2 from getting into the leaf and oxygen from getting out. And so there's this kind of game that plants have to play of balancing stomatal opening and closure and trying not to lose too much water in the process. Here is a blow-up of an individual mesophyll cell, one of these guys, and you can see how it's packed with chloroplasts. Here is an individual chloroplast, and again on this one you can see the stacked nature of those thylakoid membranes, and particularly the little space that is present inside of that membrane. So we've got the thylakoid space, the membrane, and then the stroma. We've got this setup that allows us to make a hydrogen ion gradient. The chlorophyll molecules are actually associated with those thylakoid membranes. So the more surface area you have, the more chlorophyll you can pack into that chloroplast. And then here's an electron micrograph of this chloroplast, again showing you these stacked thylakoid membranes, and then this granular stuff in here would represent the stroma. Okay, review. So which becomes reduced in photosynthesis? Carbon dioxide does become reduced, doesn't it? Is water reduced? Water is instead oxidized, right? Hydrogens are removed from it. Where do they go? They temporarily go to NADP. And then from NADP, they're transferred to carbon dioxide. So this one and this one become reduced. So where do electrons come from that ultimately reduce CO2 into glucose? The electrons come from water. Okay? We rip the hydrogens off. Hydrogens have electrons. We transfer them temporarily to NADP, and then those electrons are moved on to CO2 to make glucose. ATP is not an electron donor acceptor. Okay? Steps up on the board here. The part that's not a light-dependent reaction is CO2 fixation. This is light-dependent. This is, and this is the process by which ATP is formed. So that's going to be important something to know on your next exam is which phenomena are light-dependent reactions and which phenomena are light-independent reactions. And the most common mistake that is made is moving CO2 over here into this light-dependent reactions. It's not. Which of these happens in the stroma? This one. All the rest of them are thylakoid membrane associated. So where does our increase in mass come from? 1, 2, 3, or 4? Carbon dioxide. Okay? People used to think that the mass of growth in plants came from either the water or the soil. So there was a very interesting experiment that was done way back in the 1700s by this guy named von Helmont. And particularly, he weighed soil that a plant was growing in, and he measured how much water he was putting in, and he could not account for the increased mass by a decrease in the mass of soil or the contributions of water. Increase in mass is the result of making new material for the plant, and much of the mass of a plant is this xylem wood, which is primarily cellulose. Cellulose is a carbohydrate. So where does that carbon come from to make that carbohydrate? CO2 is being reduced into glucose, which then makes cellulose. Okay? So the mass of plants comes from the carbon dioxide that they absorb from the atmosphere. Okay, so let's dig into the light-dependent reaction in a little bit more detail. As it turns out, when you look at a thylakoid membrane, so you're looking at one of these little thylakoid membranes right here, that pigment molecules involved in photosynthesis are actually clusters of several molecules. Typically, there's about 200 to 300 pigment molecules in this cluster, and these clusters are called photosystems. So photosynthesis typically doesn't involve just a single solitary chlorophyll molecule. It involves a group of pigment molecules which actually are functioning together as a unit, and we call these units photosystems. And they gave very imaginative names to the photosystems. There are two types of photosystems. Photosystem 1 and photosystem 2. So one of the things that we're going to want to look at and see is what's the difference in these two functional units, these photosystems, and what's going on there. The pigments that you find in these photosystems do include chlorophylls, But some of the pigments are also the accessory pigments. Remember I told you last time that there were some other kinds of pigments involved in photosynthesis? There are the xanthophylls and the carotenes. So it's kind of a whole gob of these pigment molecules are organized together to make this functional unit called the photosystem. There's a special molecule of chlorophyll kind of at the center of the photosystem. And that special molecule is called the reactive center. And this is where the real kind of important stuff happens associated with the light-dependent reactions of photosynthesis. So what are all these other pigment molecules doing? Are they really just unimportant? Those other molecules are important. They're referred to as antenna pigments. What's an antenna do? This is a concept that probably most of you haven't experienced that much. Television used to be watched by a device on the top of your roof gathering the waves of energy that were put out by the broadcaster and funneling those to your television set. Now we have cable, okay? But an antenna was this kind of tree-like device that grabbed the signals and kind of funneled them toward your TV. Well, that's what these accessory pigments and other chlorophyll molecules that aren't the reactive center are doing, is they're grabbing wavelengths of light and kind of funneling it toward that reactive center. Here's a look at what chlorophyll looks like. Chlorophyll is an interesting molecule. It's really got two functional parts to it. This part in green right here is called the porphyrin ring. Don't worry about that name. It's not that important. And then attached to that porphyrin ring is this large hydrocarbon tail. This tail helps to anchor the chlorophyll molecule into that thylakoid membrane. Okay, this is nonpolar. It's nonpolar inside the membrane. And so this tail kind of fits inside this bilayer right here and holds the porphyrin ring into place. There's some significant things about the structure of this porphyrin ring. And one of the things that's very significant is there's a lot of double bonds joining carbons to carbons here. Anytime you have double bonds, then typically there is going to be some light absorption associated with that molecule. Double bonds tend to absorb light energy. So this is the feature that helps this chlorophyll molecule to absorb those photons of light. You'll also notice that kind of in the middle of this porphyrin ring is this atom of magnesium. Okay, we already talked about the electromagnetic spectrum. I introduced that a while ago. Remember, this is the totality of all of the energy that's produced by nuclear fusion in the sun. When all the hydrogen of the sun is being forced together to make helium, and some of the mass of hydrogen is being converted into energy. So we make all these gamma rays, X-rays, UV, visible light, etc. Shorter wavelengths, longer wavelengths, higher energy, lesser energy, and so visible light's kind of right in the middle here. Okay? And then this represents the breakdown of visible light from a short wavelength of 380 nanometers in this blue-purple wavelengths up to the longer 700, 750 nanometer red wavelengths. So when you did your enzyme assay, you were working down here in this 470 nanometer range of absorption. So what happens when light strikes a chloroplast? Well, it has to penetrate through the epidermis, and when it hits those mesophyll cells, it penetrates the cells and gets into the chloroplast. And some of that light energy passes through unabsorbed, and that's particularly the green wavelengths of light. And that's why the pigment looks green, is it's not absorbing those wavelengths of light. Some is reflected, also the green wavelengths of light. But other wavelengths of light, the blue, the reds, are absorbed primarily by those double bonds in the chlorophyll molecule. And that's when interesting things start happening. Oops. Where am I going here? Okay. We can measure what wavelengths of light the chlorophyll will absorb in a spectrophotometer. This isn't really a spectrophotometer. It's kind of a modification, much like a spectrophotometer, where you shine some light through a prism, breaking it up into various wavelengths, and then you shine those wavelengths of light through the chlorophyll solution in a cuvette, and then you measure the absorbance of that particular solution. So that's basically what a spectrophotometer does. If you take chlorophyll extracted from plants and throw it in a cuvette and measure the absorbance at different wavelengths, you can generate this set of curves called an absorption spectrum. And that's a lab we used to do. We used to have you grind up spinach leaves, extract out the chlorophyll, and then put it into cuvettes, and then take a reading at 400, and a reading at 410, and a reading at 420, and then plot out the absorbance readings, and you get this particular curve right here that is illustrative of the absorption spectrum of the pigments that you extract from plant leaves. So, here's the absorption spectrum for chlorophyll A. You see it's got a maximum up here, and then it drops off, particularly in these green wavelengths. And then we get another absorption maximum here. Here's a different type of chlorophyll. This is chlorophyll B, and you'll notice that its absorption spectrum is a little bit different than the absorption spectrum of chlorophyll A. Here are carotenoids. They have a little bit different absorption spectrum. They don't show the xanthophylls on here, but they have a little bit different absorption spectrum. So what's the advantage of having multiple photosynthetic pigments as opposed to just one? You can absorb more light, right? You can grab more of the wavelengths of light. Things that chlorophyll A can't effectively absorb can be more effectively absorbed by carotenoids or chlorophyll B. You're kind of covering your bases. If you want to see what effect a small modification in a molecule can have on its absorption, here are chlorophyll A and chlorophyll B molecules. In chlorophyll A, this particular functional group right here is a methyl group, a carbon and 3 hydrogens. Here it's a carbon, a hydrogen, and an oxygen. One slight variation in one functional group in this molecule can result in a dramatic shift in the absorption spectrum of that molecule. So, here's the absorption spectrum. Another type of spectrum that you can do is a type of spectrum called an action spectrum. The action spectrum is a plot not of the absorbance of light by a pigment, but it's a measure of the rate of photosynthesis, and it's plotted against wavelength. So what do you see? You see that at this wavelength right here, a little bit above 400, there's a high rate of photosynthesis. you'll see that in this region down here, in the mid 500s, there's a drop in the rate of photosynthesis. Here in the 700s again, you see an increase in the rate of photosynthesis. So an action spectrum is photosynthetic rate versus wavelength, as opposed to absorption versus wavelength. The absorbance spectrum, the action spectrum. How might we measure the rate of photosynthesis in an organism? Well, in this particular case, we're measuring oxygen production. Okay? In the old days, that might have involved counting bubbles of oxygen that are produced. Now it probably involves putting some type of a sensor, like one of those Vernier sensors, in there, and having the computer determine how much oxygen is being produced by that sensor. Well, what does this tell you? This tells you that there is a high degree of correlation between light absorption and photosynthesis. And that light that is absorbed causes photosynthesis to happen. Light that is not absorbed, photosynthesis doesn't happen. Okay? Seems like a fairly straightforward and pretty logical conclusion. But it does indicate that for this process to occur, light has to be absorbed. Why? So we can have those light-dependent reactions. Here's a great little experiment. I think one of the things that tends to happen as we develop more and more technology is we kind of develop this little sense of superiority, and we think that we're probably smarter than anybody has ever been in the past. One of the things that I think that you can really benefit from occasionally is learning something about the history of science and learning how people were able to come to these remarkable conclusions and insights based upon some pretty low-tech type of experimentation. Okay? This idea of the absorption spectrum being correlated with the action spectrum is really important in understanding photosynthesis. And this is something that we can do now with our technology. But back in the 1700s, we didn't have spectrophotometers and Vernier sensors. And yet this correlation between absorption spectrum and action spectrum was first realized hundreds of years ago. There was a guy by the name of Engelmann who did this experiment that was very elegant in design and illustrated what our technology does now. Here's what he did. He took a filament of algae and he laid it on a slide. And he took a light source and shined it through a prism. You all seen a prism before? A prism breaks up light into its various colors or bands. And he arranged it so that the light coming through the prism fell on that particular filament. So down here it's primarily blue light, purple light, blue light, green, yellow, red, etc. What he then did was he mixed together in that slide with the filament of algae some aerobic bacteria. Aerobic bacteria require oxygen for their metabolism. So what are they going to do? They're going to tend to accumulate around this filament of algae in sections where oxygen is being produced by photosynthesis. So what do we see? We see a big accumulation here. We don't see many bacteria accumulating here. We see a big accumulation here. Why? Because these colors of light are being absorbed and causing photosynthesis to occur and oxygen being produced. These colors of light not being absorbed, no photosynthesis. These colors of light. So back in the 1700s, Engelmann did this first action and absorption spectrum kind of together and got these really, really interesting results with this very low-tech type of approach. The more I hear about history of science, the more I am amazed at the ingenuity of some of our first scientists. I think in many respects we can't begin to match their brilliance of thought. So, here's where we are so far. We've got these light-dependent reactions here, the light absorption process, the electron transport, and the chemiosmosis, and then we have the carbon fixation process. Thylakoid associated, stroma associated. So what's going on when light's absorbed? Well, if we take this chlorophyll molecule and we shine some light on that chlorophyll molecule, the photons of light can be absorbed by the double bonds of that porphyrin ring. And one of the things that happens when these double bonds absorb light energy is that electrons in the molecule become boosted up to a higher energy level. In their normal energy level, they're said to be at ground state. And ground state is the lowest energy level. That is the most stable level. When photons of light are absorbed, these electrons get kicked up to a higher energy level. We call that the excited state. And up here, they're unstable. So what are unstable things going to tend to do? Get stable. So on its own, that excited electron is going to drop back down to ground state again. What's going to happen? The energy that boosted it up here is going to be released as the electron drops back down to ground state again. In what form? Well, actually, some energy is going to be lost in the form of heat. Other energy is going to be lost in the form of light. But this is not going to be the same wavelength of light as this. Some of that energy has been lost in heat. So this is going to be a different color of light or wavelength of light than this exciting energy was. This energy that's released is called fluorescence. So if we look at a flask of extracted chlorophyll from a plant and we shine ultraviolet light on there, what's happening? That UV light is exciting the electrons in the chlorophyll. They're being pushed up to the excited state, and then they're dropping back down to ground state again. And as they do, this fluorescence is being produced. And it's got this eerie kind of red color associated with it. Red is a longer wavelength of light than the photon that was exciting it. Why is it longer wavelength? Because some of the energy was converted into heat. So you'd actually, if you could measure it accurately, the flask would warm a little bit too because of the heat energy that was produced. So does this get us anything in terms of meaningful energy for cellular work? Well, heat's not meaningful energy for cellular work, is it? Nor is this fluorescence. All we've done is basically turn one type of light into another. But we haven't gained anything in terms of like ATP or reduced NADP. As long as you keep shining the light on these pigment molecules, the electrons just keep doing this. They jump up, they fall down. They jump up, they fall down. Interesting, but we really haven't gained anything. This represents a photosystem in a thylakoid membrane. And all these little green circles right here represent the chlorophyll molecules that are not part of the reactive center. Here is the special molecule of chlorophyll in the reactive center. So what are all these other molecules doing in the photosystem? They're being the antenna pigments. And they're absorbing photons of light. So maybe this one absorbs a photon. Maybe this one does. Maybe this one does. And when the photons of light are absorbed, electrons become excited. Okay? They start jumping up to an excited state. And when an excited electron hits another chlorophyll molecule, it kind of bumps it and it excites its electrons. So basically what you get is kind of a chain reaction occurring, where one chlorophyll molecule excites another, excites another. Same thing could be happening over here. This one might be excited by a photon. And then that excites this one, that excites this one. Okay? It's kind of like taking pool balls on a pool table. Your cue ball that you strike initially would be the original photon. When that hits another ball, it imparts momentum to it. That runs it into another ball, imparts momentum into it. And so you get this chain reaction where all the balls start moving. So what happens? These excited electrons all start kind of moving around. And ultimately, one of these excited chlorophyll molecules stimulates this reactive center. And now its electron gets boosted up to a higher energy level. What's so special about this reactive center is that electron doesn't just fall back down to ground state and produce fluorescence. Something special starts happening in this reactive center that gets us what we need to get out of photosynthesis. So you see the role that these particular accessory pigments and other chlorophylls play is they're just grabbing all those photons of light. Each one of these has a little bit different absorption spectrum, so they're grabbing all these different wavelengths of light and kind of funneling them toward that reactive center where something special is going to happen. Okay, that's probably a good place to stop because the next thing we need to do now is to see the interaction that takes place between two photosystems in the thylakoid membrane, photosystem 1 and photosystem 2. So we'll pick up here on Thursday.